every generation of each and every species resembles its ancestors in many ways. How are these characteristics transmitted from one generation to the next? This is called heredity. Your parents pass their genetic information to you, and that's the reason you resemble them. If you go into much more scientific explanation, your parents pass chromosomes, which are made up of proteins and nucleic acids. In this video, we're going to talk about nucleic acids in depth. Not how heredity works and all that, because that's related to biology. But we're going to talk about chemical composition, the structure of nucleic acid, etc. More chemistry related stuff. Okay, so there are two types of nucleic acids. We have the DNA and the RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. This is how DNA looks. And this is the chemical structure of DNA. We will learn about this chemical structure a bit later, but for now you need to know that DNA is made up of two strands, and both of these are held together by hydrogen bonds between the bases. Nucleic acids on complete hydrolysis give phosphoric acid, a pentose sugar, and a base. A base is just a compound containing nitrogen. So first we get phosphoric acid, and that's pretty straightforward, just a phosphoric acid. And then we get a pentose sugar. A pentose sugar simply means a sugar with 5 carbon atoms, because pent means 5, like pentagon means 5 sides. On the left side we have an RNA pentose sugar, and on the right side we have DNA pentose sugar. The sugar moiety of RNA is beta D ribose, and sugar moiety of DNA is beta D2 deoxyribose. Why is the sugar moiety of RNA beta D ribose, and why is the sugar moiety of DNA beta D2 deoxyribose? Well, if you take a look at the Fisher diagram of ribose, it looks something like this. And since the OH group is on the right side, we know that it is the D configuration. So since we have a ribose and the D configuration, it is called D-ribose. But when you convert it into a Harvard projection, it looks something like this. And since the OH group are upwards, it is beta configuration. If the OH group were down, then it is alpha. But in this structure, the OH group is upwards. So therefore, it is a beta configuration. So we have beta D ribose. So beta means the OH group is upward, and D means the OH group is on the right side in the Fisher diagram. So we got beta D ribose. You can see that the structure of DNA is exactly the same as structure of RNA. But the thing is, in this side you can see we do not have an oxygen. So in RNA we have OH, but in DNA we have just H. So we do not have an oxygen. That's why it's called deoxyribose, because deoxy means like no oxygen. So that's why it's called a deoxyribose. And it's called 2-deoxyribose because in the second position of carbon atom, oxygen is not present. We will learn the numbering of the carbon atoms, but this is the first carbon atom, and this is the second carbon atom, and this is the third carbon atom, etc. So in the second carbon atom, there is no oxygen attached to it. Instead of OH group, we just have an H. So that's why it's called deoxyribose or 2-deoxyribose, because in the second position, instead of OH, we have H. So O is removed. And then we have the base. There are 4 types of bases: adenine, guanine, cytosine, and thymine. So these 4 bases which I just mentioned are the bases of DNA. RNA also has bases. The first 3 bases of RNA are same as DNA. So the bases of RNA are adenine, guanine, and cytosine. But instead of thymine, we have uracil in RNA. So the 4 bases of DNA are adenine, guanine, cytosine, and thymine, and the 4 bases of RNA are adenine, guanine, cytosine, and uracil. So instead of thymine, we have uracil. So on hydrolysis, we get 3 things: a phosphoric acid, a pentose sugar, and a base. So it means that these 3 items are present in nucleic acids. And yes, they do. So let's learn how these 3 items are structured in a nucleic acid. First of all, let's take a pentose sugar. The carbon atoms are numbered from 1 to 5 because there are 5 carbon atoms. So each of the carbon atom has its own number and it's denoted by 1 dash, 2 dash, 3 dash, and so on. When the first position carbon atom, that is 1 dash carbon, is connected to any base means like adenine, guanine, thymine, cytosine, etc. So if the first position carbon atom is connected to any base, it's called a nucleoside. So this is a nucleoside, a pentose sugar connected with any base. And that base is actually connected to the first position carbon. And in a nucleoside, if the fifth position carbon atom is attached to a phosphoric acid, it's called a nucleotide. So nucleoside means the first position carbon atom is attached to a base, and nucleotide means the first position carbon atom is attached to the base as well as the fifth position carbon atom is attached to phosphoric acid. So that's a nucleoside and a nucleotide. But this is not the complete structure of DNA yet. It's just the structure of nucleotide. So we learned the structure of nucleotide, not the complete structure of DNA. A DNA is formed when multiple nucleotides combine together. So we have a pentose sugar, a base, and a phosphoric acid. Two nucleotides will combine in this manner. The fifth position carbon will have a link with the third position carbon of other nucleotide. So as you can see, both of these nucleotides are now linked and they are called dinucleotide because di means two. So two nucleotides are combined together or joined together. So they are called a dinucleotide. So this is how a dinucleotide is formed. Two nucleotides are joined together with phosphodiester linkage. So the linkage between them, so how they are joined, so that's called a phosphodiester linkage. Let's simplify this diagram a little bit. So we'll make it like this. So the circle is the phosphoric acid, the pentagon is a pentose sugar, and this shape is any base, so it can be any one of the 4 bases. So two nucleotides will be joined together in this manner. So we have discussed already. So this is the phosphodiester linkage. So it's not necessary that only two nucleotides should be combined together. We can have multiple nucleotides combined together in a long chain. So if we go back to our previous diagram, like in the starting of the video, we saw that the diagram of a DNA was like this. So this is actually a long strand of nucleotides. So you can see all of these nucleotides are joined together in a long chain. Since nucleic acids are formed when multiple nucleotides combine together, that's why they are called polynucleotides. Okay, so we have talked that multiple nucleotides will be combined together in a long chain. And this long chain actually is a primary structure of DNA, or a nucleic acid actually, to be general. So primary structure talks about how all these nucleotides are linked together in a long chain. We also have a secondary structure. In secondary structure, we learn how bases form bonds between each other. So we have a primary structure talking about the long chain of a nucleotide, and we have a secondary structure talking about the connection between bases. So now in secondary structure, we have something called a double-strand helix structure. This structure was given by James Watson and Francis Crick. In this structure, the DNA looks something like this. So this is what comes in your mind when you hear DNA probably. So this is actually a double-strand helix structure of DNA. So you can see we have a long chain of nucleotides. We have one chain and two chains. So we have 2 chains in total. Now in these 2 chains, we have the bases obviously. So these bases will form hydrogen bonds between them. So we have 2 chains of nucleotides or 2 primary structures, and then the bases form bonds between each other. So this actually is the secondary structure. Let's take 2 chains of nucleotides, each of them anti-parallel, or in simple words, just going opposite to each other. So you can see in the first chain, it starts with the 5th position carbon atom and ends with 3rd position carbon atom. And in the second chain, since it's opposite, it starts with 3rd position carbon atom and ends with 5th position carbon atom. So for the sake of simplicity, again, we're taking red as cytosine, blue as guanine, orange as adenine, and yellow as thymine. Cytosine always forms bonds with guanine, and adenine always forms bonds with thymine. So adenine does not form a bond with guanine or cytosine, and guanine doesn't form a bond with thymine. So always cytosine and guanine form bonds, and adenine and thymine form bonds. So it could be a cytosine guanine bond or a guanine cytosine bond. It could be an adenine thymine bond or a thymine adenine bond. In any case, adenine always forms bonds with thymine, and cytosine always forms bonds with guanine.